Hello guys. Today we're going to talk about biomolecular metabolism, and basically what we're going to discuss today is how do you build and break down molecules that you need for everyday life. All right, so we're going to talk about chemical reactions that power everyday living. And so by the end of today, you're going to be able to describe the properties of monomers and the types of bonds that connect the monomers in biological macromolecules to build polymers. And explain the energy changes involved in these two different processes. All right. And so you want to think about what are biomolecules and why do you think they are important for maintaining homeostasis, a balance in living organisms? And if you don't know what the word biomolecule is, maybe you've heard it as macromolecule, right? Maybe you've heard of macros when certain people want to hit certain macros in a day. So what do you think they are? So think about it and share your answer. And we want to remember that our macromolecules that you find in your food, right, are carbohydrates, lipids, proteins, nucleic acids. We have four essential macromolecules. And macromolecules are basically organic and they are built up of carbon. All right, sorry. And so why is carbon so important though, right? Carbon is important because it has 4 valence electrons that can be used for bonding. And so therefore, because it has 4 valence electrons, which you can see that are represented in blue, it can always covalently bond. It can form 4 covalent bonds. What a covalent bond is, remember, is that it's the sharing of electrons. So here you can see that the red is representing the electron from the hydrogen and blue is representing the electrons from the carbon. You see how it's sharing? So this is representing a covalent bond. Do you guys remember what type of bond water has? And so water does have covalent bonds, but you guys want to remember that water has covalent bonds within each water molecule. And water also has hydrogen bonds between water molecules. So you want to remember that key difference. When you're thinking of water, you're thinking of the fact that the bonds within a water molecule are covalent bonds. So between the oxygen, hydrogen. And then the bonds between water molecules are hydrogen bonds, okay? So hydrogen bonds form between different water molecules, right? I just wanted to clarify that. And I know it was one of your questions for the warm-up, for the true or false. All right, and so, carbon is very important because it can form up to 4 covalent bonds. And like, for example, hydrogen can only form with, have 1 bond. Oxygen usually forms 2 bonds. Nitrogen usually forms 3 bonds. And so carbon-based molecules can form a variety of different structures. And it's because of the ability to form 4 bonds, okay? And so because it can form up to 4 bonds, then it means that there's more available structures, which means that it's more available functions, and it can build more complex life forms, and therefore there could be a formation of more species of life, right? And so remember that structure determines function in molecules, and that's why the more structure available, then there's more available functions, right? Because structure determines function. You always want to remember that. Alrighty. And so what is a biomolecule exactly? They're organic, meaning they're carbon-based molecules, and they can be used to be converted by living things, okay? Oh, sorry, for this section, I wanted to talk about the fact that cells have 72% water. Remember we talked about the properties of water video and how water is super important to maintain homeostasis within a living thing. And the other 25% is carbon compounds, which are carbohydrates, lipids, proteins, and nucleic acids, which are the biomolecules or macromolecules, same thing, that we need for to live. And the rest of the 3% is sodium, chloride, potassium, etc. So salts. All right, and like we said, organic is carbon-based molecules. It can be used and converted by living things, and we all need all living things need biomolecules. Again, these are the 4 types of biomolecules, and here you can see a little animated picture of each of them. Carbohydrates, you're going to need these for quick energy and structural support. Lipids are for long-term energy storage and cell membranes. Proteins, you have different types of proteins. And an example of a protein could be an enzyme. We're going to talk about the importance of enzyme in this video. Proteins could also be used for structure. They can be used to transport things within a cell, for defense, for signaling. And nucleic acids, you need these for genetic information storage and transfer. All right. Wait, sorry. Give me a second. My cat wants to get it. Alrighty. And so let's get into some terminology. We talked about the fact that these things right here, they're called polymers. And basically what polymers are large biomolecules made from smaller building blocks called monomers. So here you could see the carbohydrate, it has repeating monomers. The protein has repeating monomers. And nucleic acids also have repeating polymers. All right. So what is a monomer? What is a polymer? A monomer is basically a single building block. So like, for example, one Lego brick. A dimer is 2 monomers joined together by covalent bonds. And the polymer is many monomers that are joined together by covalent bonds. Okay? And so how are monomers built into polymers, right? And so we're going to talk about something called dehydration synthesis in a second. And so how do you break down polymers into monomers? Then we're going to talk about something called hydrolysis. And both of these reactions need enzymes. Okay? And so like I said, dehydration is the combining of monomers to make a polymer. So here you can see that these 2 monomers are combined to make a polymer. And so what do you need for dehydration synthesis? You need 2 monomers approaching each other. And you're going to need something called an enzyme in order to facilitate the reaction by lowering the activation energy. So it means that you're going to need less energy for the reaction to occur. So it's going to facilitate it. And the hydroxide from one monomer and the hydrogen from another from the water that's being released. Okay, so during the dehydration synthesis, you're going to be releasing water as a byproduct. Okay, and so water is removed and covalent bonds form between the monomers. So here you can see a covalent bond. And the process is repeated to build a long polymer chain. So here you can see your polymer chain that is covalently bonded. Alrighty. And this right here, OH, is what we call a hydroxyl group. And H stands for hydrogen. Again, you need enzymes to facilitate the reaction and reduce the activation energy. Alrighty, so I'll show you a video of dehydration synthesis. So in dehydration synthesis, remember, you're going to be using water as a byproduct, making water as a byproduct, and then bonding, covalently bonding to monomers. So here you can see 2 simple sugars creating 1 complex sugar. All right, so again, monomers are covalently bonded together into polymers. And so let's get into more specific terms. So dehydration synthesis is called an anabolic process. So it's building up larger molecules. And it's going to synthesize or create polymers for energy storage and or structure. So the whole goal of making things is either to store energy or for structure. And so in this case, because it's a sugar, then it's going to be to store energy, right? And like we mentioned, you're going to need enzymes for assistance. So it's going to help make the reaction more, it's going to facilitate the reaction by lowering the activation energy. And so dehydration, again, you're going to have water as a byproduct. So you're removing water. And synthesis means to create something. The bond you're making is a covalent bond. So to sum it up, dehydration synthesis is the removal of water to make a bond, covalent bond between monomers. All right. Again, water is formed as a byproduct. All right. So we talked about dehydration synthesis, which is using monomers to make polymers. So how do you break those down? All right. Oh, sorry. Yeah. Dehydration, we talked about that. All right. So hydrolysis, it's the opposite. It's the breaking of polymers into monomers. You still need enzymes. All right. You still want to facilitate the reaction. You want to make it easier for the chemical reaction to occur. You're going to use something called an enzyme, which is a protein. Water molecules are going to approach. So you need water, the reaction, and it's going to split the hydroxyl, which is, hydroxyl, remember, is OH. It's going to split, water is going to split, into hydroxyl, going to one monomer, and hydrogen going into another monomer. So here you can see you start off with one hydrogen and one hydroxyl, but here the water is going to split into a hydroxyl and into a hydrogen, to two different monomers. Then you're going to continue the process. But before you continue the process, you want to remember you're breaking down these covalent bonds. So you're going to continue the process until you break down the entire polymer. So here you can see you have single monomers, each with its hydroxyl group and hydrogen. And you no longer have any covalent bonds, okay? You're no longer having any sharing of electrons. All right, and so let's watch a video of complex sugar, which is a disaccharide being broken down into simple sugar, so monosaccharide. So here you see that water was needed, and therefore two monosaccharides are formed. All right, and so again, this is the process by which polymers are broken down into monomers. We said that dehydration synthesis is an anabolic reaction. Hydrolysis is a catabolic process, all right? So it's going to break down polymers to release energy. And the way I remember this is I like to think like, okay, my cat likes breaking things. Just a quick way I remember it. So it's the breaking down. And it requires assistance of enzymes still, just like dehydration synthesis, in order, and the breakdown of water to occur. So you need water. Hydro is water, and lysis means to break a bond. So hydrolysis uses water to break the bond between monomers. Okay? Lyse, I always think of it like lyse. You're lysing something, it's breaking down something. Hydro, you need water. All right? All right, so think about this question. Which one do you think is a monomer and which one do you think is a polymer? Do you think that this is dehydration synthesis, or do you think this is hydrolysis? All right, and so this one is dehydration synthesis. You could see that you're going to combine two monomers to make a polymer. All right. Synthesis meaning to create. So you're not lysing, you're not breaking anything, you're creating something. And dehydration meaning you're gonna have water as a byproduct. You're removing water, right? So here you see your monomers combining to make a polymer. Alrighty, and so for example, if you had 10 monomers, so you could have your fingers near each other. So those are 10 monomers, and make sure your thumbs are near each other. How many water molecules do you think that you're going to need to completely hydrolyze a polymer that is 10 monomers long? All right? How many water do you think you need? Alrighty, and so if you have your 10 fingers next to each other, even your thumbs, you want to count the bonds within, so all the spaces. And if you were to count, so from your pinky to your ring finger, that's 1. From your ring finger to your middle finger, that's 2. From your middle finger to your index finger, that's 3. From your index finger to your thumb, that's 4. From your 2 thumbs, that's 5. Thumb and index 6. Index and middle finger 7. Middle finger and ring finger 8. Ring finger and pinky 9. So you have 9 bonds, right, between. So then you're gonna need 9 water molecules in order to create a polymer. All right, so that's how you solve it. You figure out how many bonds are connecting them, and then that's the amount of water that you need. Okay. And so the general rule is for the amount of monomers, just subtract 1. So you have 10 monomers, so just subtract 1, and you're going to need 9 water molecules in order to complete your dehydration synthesis. All right? All right, and so here you could see that. So you need water for each bond. All right. Before I move on, I want to talk about why enzymes are so important. I don't have a slide for that. But basically, if you remember, you need enzymes for both dehydration synthesis and also for hydrolysis. And so enzymes are going to be useful to break things down or to create things. So enzymes can either be useful to break things down or create things. They're a type of protein. And so usually biological reactions occur very, very slow. And so without enzymes, it wouldn't be possible because enzymes lower inactivation energy. And so the energy barrier to start reaction. So you're going to need less energy than if you didn't have an enzyme. And so enzymes are not consumed. They can only be reused. And so you call enzymes a catalyst, something that can speed up a reaction. All right. When we talk about our protein lesson, we're going to talk about structure and function. And enzymes, the temperature and pH and concentration does affect whether or not it's going to work. So it does affect its function. But we'll talk about that in the other lecture. All right. And so let's move on to the biomolecular metabolism. So we have metabolism. What is metabolism? It's the combination of chemical reactions that synthesize and hydrolyze biomolecules for energy storage and release in an organism. All right. So you have anabolism, which is going to build up. It's an endergonic reaction. All right. And then you have catabolism, which is going to break things down and is an exergonic reaction. All right. Exergonic means that you're going to release energy, and endergonic means that it's going to require energy. Okay. Endergonic, you're going to release energy, and exergonic means that it's going to release energy. Okay. Alrighty. So catabolism versus anabolism. All right. So let's compare them. So free energy is energy that is actually available for a cell to use for a metabolic process. Catabolism, again, is the breaking down of biological polymers into monomers to help generate ATP. ATP is basically just the energy source our cells need. And again, it's exergonic, okay? It's going to release energy. Anabolism is going, reactions are going to build up monomers into polymers for energy storage, okay? And so anabolic reactions are endergonic, meaning they require energy, okay, into the bonds between the monomers. And so exergonic reactions release free energy that is used by endergonic reactions, so they can occur. So basically, when you have your exergonic reactions, It's going to release energy, right? We said it's going to release energy. And then it's going to be used by your endergonic reactions. So your endergonic reactions are relying on the release of energy in order for them to occur. And this is called reaction coupling, right? They rely on each other. And here you can see the little graph. So the amount of energy, you're going to release it. And here you're going to need energy, right? So it's going to start off a little slower. All right. And so again, I'll talk about energy coupling a little more. Exergonic reactions, we said exergonic reactions over here are going to release free energy. So you could think of this like rolling a ball down a hill. You don't need much energy for this to occur, right? Endergonic reactions, you're going to need energy. So you're going to think of like pushing a ball up a hill. So you were to imagine if you had to push something, you'd need more energy, right? And so here, if you're throwing down a ball, you don't need much energy for it to just fall, right? It's going to fall naturally. Energy coupling is a biochemical process where the free energy released from an energetically favorable reaction, such as exergonic reaction and hydrolysis of ATP, is to power an energy unfavorable reaction, which is the endergonic, right? Alrighty. So before I get into the main topics, I thought I had a picture. Give me a second. All right. And so here you could see, like, for example, in ATP hydrolysis, which is an example, you're going to want to need, you're going to have an exergonic reaction, which is going to provide energy for protein synthesis from endergonic reactions. So this is like using energy from a waterfall to power a factory. All right. So you need energy from exergonic reactions in order to have endergonic reactions. Okay. And so here you can see that in ATP hydrolysis, water is being released, energy is being added in order to create something. And then here you have hydrolysis, which you need water, and you're going to release energy. Okay, so it's a chain. This is energy coupling. And you see this in ATP hydrolysis. Okay. Alrighty, and so we want to remember that free energy is energy that is released to do work in a cell. Exergonic and endergonic reactions are coupled together. Exergonic reactions release free energy that is captured and used to make endergonic reactions available. You could think of this like energy is exiting, it's releasing it. You need energy to enter in order to occur. Dehydration synthesis is an anabolic chemical reaction that forms covalent bonds between monomers to build polymers. So, for example, water is formed as a byproduct. Dehydration, remember water being released. Synthesis, meaning you're creating something. Hydrolysis is a catabolic chemical reaction that breaks the covalent bonds between monomers. Remember, catabolic, think of your cat breaking things, breaking covalent bonds in order to break down polymers into monomers. And you need water. So hydro meaning water, you need water. And so both dehydration synthesis and hydrolysis, again, require the assistance of energy to occur in living organisms.